Hi, will you let me swab your mouth? Yeah. Oh, thank you.
Ready, kitty?
Ew, we're just gonna be really quick, okay? Yeah, I'll be fine.
It'd be good.
Hey, can I get in here? Please. Hi. Hi. Oh, that's such a good girl. Can I get in your condo? Let me in there.
Please, Illy, come on.
Yay!
She's like, put me down.
You upset because I swabbed your mouth? You got a lot of bacteria in there, so.
Alright, so this is our final sample going in the incubator. Here are all of our t-shirts and such. Looks like we've got some rows, that's exciting. We'll leave them until later in the day and then we will grandstand them. Yeah. Oh, gross. Some
I'm not in their group. Don't get it twisted. Watch me be fine.
For some of the animals, we believed that their specimens were actually Staphylococcus species. So for those, we decided to plate them onto mannitol salt agar plates. This allowed us to differentiate between the species that fermented mannitol and those that did not. None of our plated specimens actually fermented mannitol, so we concluded that none of our Staphylococcus species were pathogenic. For nuggets in a rotube, we indicated positive glucose, positive lactose, positive urease, and positive citrate activity. Based on these findings, as well as the gram-negative status of the bacterium, we concluded that our best guess would be that the bacterium species falls somewhere under Citrobacter or Klebsiella genuses. For Jackson's inner rotube, we came back with variable glucose, negative indole, and positive citrate activity. Based on these findings, our best guess is that Jackson's unidentified bacteria was Bacillus cereus. This was also based on the gram status of Jackson's bacteria. Finally, Holly's inner rotube came back with negative indole, positive citrate, variable urease, and variable adonitol. Based on these findings, as well as the gram status of her unknown, our best guess for Holly's unidentified bacterium is Pseudomonas fluorescens.
Hey friends, Andy here. I just wanted to take a minute to kind of share with you all in a little bit more detail what this vlog was really about. So this aerobic bacteria, cats and dogs' mouths, has actually been documenting our being mine and Megan's project for the lab portion of our microbiology course. So the whole Jam by Cam project is part of the microbiology course, and for the lab portion of that we had to develop a project, and Megan and I and 2 other members of our lab group chose to do this project focused on looking at the bacteria that we could get to grow from dogs and cats' mouths. And our initial question, in addition to just, can we get something to grow, and if so, what is it that grows, was also comparing dogs' mouths versus cats' mouths. So which ones, whose mouths, dogs or cats, were actually cleaner. Dirty being defined as more pathogenic or more potentially pathogenic species. So in conclusion, our results pretty much showed that there were potentially pathogenic species on both sides, cats and dogs. Altogether, the cats showed a wider variety of potentially pathogenic species, so I believe like 6 out of 9 of the cats plates that we grew and took to further testing and gram staining ended up being potentially pathogenic, and only like 3 or 4 out of the 9 dogs were potentially pathogenic. So these results are very inconclusive based on the amount of time that we had to complete the project, as well as the means, the resources that we had available to us. We really weren't able to do all of the testing that would need to be done to make more conclusive, or to get more conclusive results. We can't say for certain that anything that we found is actually pathogenic. We're literally just going off of what we think could potentially be pathogenic. So all in all, it was a really fun project. It was really cool. Megan and I both have a public health background, so from a public health standpoint, looking at what is in the mouths of our pets, our cats and our dogs that are oftentimes living in our homes, a lot of times kissing us on the face, what is actually in their mouth. So from a public health standpoint, it was a really cool project. We had a lot of fun working with Courtney and Haley. They were an awesome group, and a really great addition to the Damn Guy Cam family. So I really hope you guys enjoyed this vlog. If you have any questions, let us know. If you have any suggestions for our project, definitely leave them in the comment section below. Bye!